Yo, and welcome to Shooting the Soil. Today we are looking at a 15-gallon indoor pot. This is the third sample we've done. Check the description for the previous samples. And what we have recently added is some mycorrhizal inoculant. So we are hoping to see more fungi and some spores and stuff like that in this sample. Anyway, my name is Matt. I'm currently a student of Matt Powers, studying regenerative soil, working on my certification. And we are sharing this microscopy with you guys to help everybody get a better understanding and a better feel for looking at our soils under the microscope, because it's all about the biology. Alright, so already I'm seeing two fungal strands just here in the top corner. This one's a nice brown color. We're going to search around a little bit more before we take some closer looks at those. Let's just get square here with the camera. Alright, what else we see? Huh. Interesting. Didn't see these. I almost wonder if it's kelp, because I did add a little bit of kelp top dressing. But all right. Well, while I'm looking around here, feel free to ask any questions in the chat if you would like. So right here we're just looking at a little piece of a leaf, I should say, organic matter. And alright, scrolling on, see if we find any microorganisms, any big ciliates or nematodes. Got quite a few air bubbles. Could have done a better job dropping the slide properly, but that's okay. Alright, let's see where we're at here. Don't see anything big moving around yet, but seeing quite a few little strands. More organic matter. Alright, we're over to the edge now. Down a little bit, and we'll go back across. And that might just be a thread or something. It got stuck on the slide, probably on the slide cover. Alright. Get a little bit more in focus. Definitely seeing more strands of fungi. A lot of big air bubbles, though. But good amount of aggregates. Still got a lot of clear ones. Hoping to get those all aggregated and starting to break down by the biology. But it takes time. Looks like another good... That looks like a fungal hyphae there and there. Yeah, why don't we just take a quick zoom in? We could zoom back out and repeat our scan, but we are at 100x right now, and now we're going in to 400x. Let's get focused in here. Oh yeah, I could probably add a little bit of shading. Get our aperture dialed. And that's a kind of weird looking strand right there. Let's see, where is that other nice straight brown one that we saw? I thought it was right above. Yeah, right there. That's a nice colored fungal hyphae. That brown humic color. Yeah, nice septums. You can see the separations. So that's a good fungal hyphae. Thought there was a bigger one right around here somewhere. Anyway, while we're in this close, let's just take a look at the bacteria and see where we got a lot of round ones here, but that kind of looks snake-like. Could be a spirilla. Let's see, where do we see a nice pill-shaped? It's kind of pill-shaped. one over here, but not a really good, I don't see a really good example of a basilisk. Usually I see one in just about every frame. Let's see what we can find here. A lot of little critters bumble around down there. But, another one there. Might need to add a little bit more EM. We're seeing some rotifers in there. They might be going to town and eating all my bacteria. But let's see. You can find a good lactose basilisk. Oh, that's an interesting. So there's a fungal spore. Forget what type that is. But here's some more pill shaped. Oh, that looks like a. What's that moving around up there? See if I can get a little bit better focus on that. That's about the best we can do, but yeah, that kind of looks like a spirilla bacteria. Another little snaky one there. That's kind of basilisk shaped. So yeah, diversity is the key. More shapes and sizes, the better. So yeah, still pretty healthy bacteria amount. Yeah, see there's a little pill-shaped one. That's probably a basilisk. And the round ones again, cocci, coccus. Alright, let's actually go back out to 100, continue our scan. See if we can find some bigger microorganisms. I think I was moving this way before. Oh, there we go. What was that? Whoa, that was fast. That was a ciliate. I don't know if you caught that. Where'd he go? Whoa. Holy cow, that one's fast. I might have lost it. Yeah, but if you caught that round blur flying across my screen, that was some sort of ciliate. Hopefully we run into him and he's not moving so fast. And we got some more good, nice good looking strand right there. Oops. What am I knocking around here? I'm knocking around my filter. Alright. Let's finish our scan across. Oh, yep, that's to the edge. Let's go down. A whole bunch of these fungal hyphae there, a little piece there. So, definitely an improvement. Oh, I saw one of these before. It's rare that you find something perfectly round that's not living. So that's either a fungal spore or a cyst. This is moving around quite a bit. Is that in a... Yeah. Well, let's go in a little closer to 400x and see what, see if we can get a better look. So yeah, here's that. Oh. Not quite sure if it's a spore. Like, this is definitely a little tiny fungal spore right there. So this is quite large if it was a fungal spore. So it looks like there's a bunch of stuff in here. I bet that's some kind of microorganism cocooned up in a cyst. It's almost like you can see something here, but I think that's just the top of it. Pretty fascinating looking. I don't know what's moving everything over here. I mean, keep on looking over there, but I think it's just all the bacteria around it. Just bumbling about and moving that piece of silt or sand. Alright. Anything else cool? Oh wow, another really good Cool looking fungal spore starting to sprout. Yeah. Let's get more fungi in there. That's what I need right now to balance out my ratios. Yeah, that's probably a nice little basilisk there. Quite a bit of bacteria. Alright, let's whip back out to 100x, see if we can find some cool looking critters to look at. I'm gonna find a nematode. I have found them in my indoor pots before. Hopefully we can find one. Oh, up to the edge. A lot of air bubbles in this slide. Sorry about that. It's an interesting looking strand right there. Hopefully that's beneficial. Alright, we're to the edge. Had uniform sides and everything. That strand up there anyway. Alright. What else cool can we find to look at? Let's see. Well, yeah, right here is nice aggregation. That's what you want to see in your soils. Huh, what's... This is very interesting. I'm not sure what this is. It almost looks like it could be an amoeba, but that color is very unique. Could just be another fungal cyst. Well, why not? Let's pop into 400. Take a better look at that. What might you be? Very large fungal spore. You know, like this is definitely a fungal spore. But what the heck is this thing? Doesn't look like an amoeba at all. Guess it could just be organic matter, but perfectly round like that, I doubt it. Well, flip back out and continue scanning around. See if we can find anything else cool. Alright. Well, really nice strand here. Alright, yeah, we gotta go in and look at the nice long fungal hyphae. Oh yeah, that's a good one. Wow. And I think that's the edge of it. Yeah, but see how it's nice and equal, uniform. You got the septae, that's the separations in the cells. Yeah, this is one of the best fungal hyphaes I've seen in here. For the longest anyway. It's probably at least, I would say, 4 microns wide. That's nice. It's going to provide some good nutrients for my plants. Alright. Flip back out, continue the scan. Yeah, wow. It's a pretty nice fungal hyphae right there. Oh, we got another really dark one. Yeah, definitely, you know, every other frame seeing fungi hyphaes now, where before I really had to search for them. So that's a good way to tell that you're improving and advancing your soil. Alright, let's scroll back down. Back across, hopefully spot a nematode. Organic matter. Kind of looks like an amoeba right there. But let's keep scanning around. Fix the focus. Alright. Yeah, pretty good aggregate level and everything. Another air bubble. Oh, got a little fungi hyphae in there. That looks more like organic matter. Oh, knocking the slide around. Alright. Anything cool swimming around? Alright, we're at the edge. We got about 2 more passes back and forth before we scan the whole thing. We are at 100x right now. Phenotype. So we got that's the 10x objective. And then with the magnification and that would be the eyepiece, but is my camera, it equals right around 100x. Probably a little bit more than 100x than you would normally see. And once we find something cool to look at, we'll dive into 400x. And we can even go into 600 if we want. Oh, thought that was motion, but it's just the slide drying. Alright, let's go down to right about here. Do one more scan across. Oh. Skinny long hyphae. Alright, we're going to have to... And another one of those round brown things that we looked at before. It's the second one we've seen. Thought I just saw somebody swimming on the outside. Well, we got to get moving before the slide dries here. Oh yeah, a couple more hyphaes. One in the top there. Would be a piece of a root there. Alright. Some more hyphae sticking out of this aggregate. And go over to the side. Yeah. Take one more pass around, see if we can... There's a big fungal hyphae segment. Just kind of panning around now, seeing if I missed anything. Oh, yeah, there's another one of those round, weird looking brown things. Not sure if it's a cyst or a very large spore. Interesting though. Kind of wanted to... I was hoping we'd spot that super fast moving ciliate again. I thought I just saw something move. I'm using this microscope right here. Is not that expensive. It's only a Swift 380T. They're right in the $300 range, $350-ish, I think. And another good one is AM Scope. They also make a pretty good inexpensive microscope. Yeah. That's a nice fungal hyphae right there. Yeah, let's drop into 400x one more time. Just check out this one. We can find where we are at. Where was it? I lost it. Thought I had it in the center. Shucks. Hate when that happens. Back out to 100. There it is. Alright. That's right. That should be pretty centered. There it is. Yeah, so this one's probably, I would say, 5 microns. Nice big fungal hyphae. Nice and uniform. Definitely beneficial. Whole bunch of bacteria down there in this frame. I was just moving really fast, but don't think it was so silly yet. What's moving all this around? Just bacteria? I think so. Yeah, not seeing any fragilates or anything like that down there. But no, I just, I don't really sift out that sort of thing. They kind of, well, you have this, the eyedropper. So, the eyedropper is not going to pick up, a rice hull or anything like that. But you just kind of want to do a dilution with water. I do a 1 to 5. And usually the drops come out, without too much big sediment in there. I did have to redo this slide right before we started the stream because a larger piece of sand or something was on the middle of the slide, and then the slide's sitting at an angle, and every once in a while you got to redo it. And they make better droppers than that. I think I have one somewhere over there, but yeah, you got a good pipette and put 1 drop out. It's usually not gonna contain sand, and if not, you just wipe it off and start over. Alright, let's see. Anything else cool? Well, we might finish off by just going out to 100, and actually we can switch over to the dark field just because it looks really cool, and maybe scan around just a little bit. So put in our dark field filter, and let me switch over to manual mode, and I just gotta open my light. Bring that up, and there we go. And now we are in the dark field. It's an interesting way to look at things because all the humic, like the humic acids and what the bacteria is releasing to break down all these minerals sort of glows orange. So you kind of get a really good feel for your aggregate level. Otherwise, if you looked at, just sand or clay, all you would see is this clear stuff. Alright, so we'll take a quick little peek around here while we're in the dark field. Actually, we could use a little bit more light. Oh, wrong way. There we go. Okay, a little bit more focus. Yeah, there we go. That's looking a little bit better. I thought I just saw something move. And another cool thing about the dark field is, this is probably I'm pretty sure this is a fungal hyphae. They sort of glow, or phosphoresce, I should say. They get kind of like rainbow colors as the light passes through the middle of them. So that's like another way to kind of identify that, oh, yeah, yep, that's a living fungi. A living hyphae. Alright. Yeah, so just scanning around right now. Feel free to ask any questions before we we sign off on this sample, but all in all, I would say seeing improvements. It's getting the soil is getting better and better. Seeing more fungi since I added the hyphae. Seeing more shapes and sizes and diversity of the bacteria with the EM that I added. That was in sample 2 video, which I got in the description down below. And then if you go back to sample 1, there wasn't I mean, it was looking pretty good, but not quite as good as this. Not as much hyphae. Not sure what that is. That's really interesting. It's kind of like bow shaped. Let's see. This soil is probably, this is in it's halfway through its second run, so it's just about a little over a year old. So first run of plants through it came out excellent, and we amended with just topsoil amendments, worm castings, some oyster shell, put a little kelp on there, and then we've just been adding compost extracts and a little bit of EM, and other than that, just watering. And so far, halfway through the second run, plants are still looking very, very healthy. Hopefully, I'm just going to keep on making the soil better and better just by getting the biology right in there. That's really interesting looking. We got we got some what looks like to be a fungal hyphae there. And whenever you see this crystallization, it always fascinates me. Because what I'm learning in my regenerative soil course is fungi digest externally. So sometimes you can kind of see like clusters of like sugars or nutrients kind of crystallizing up on the outside of them. I'm not sure if that's what we're looking at here. Trying to go through the focus. Oh, somebody see there was a look at that little bumbling guy. Let me get the mouse out of the way. But that was a little fragilate flying around. Small, fast protozoa. Yeah, that's interesting looking. Kind of want to switch out of go back to auto here. And before we sign off, just take a look at this in the bright field. Come on, get out of their dark field filter and see what this looks like in the dark field. This definitely looks like a fungal hyphae. And then, yeah, I was hoping this looked more like a root, but it just looks kind of like chomped up, decaying organic matter. Let's see. Let's throw a little shade in there. Go into 400x. Yeah, interesting looking. Like, it does look like some sort of, crystallization or something on the outside of that strand. I haven't heard of ghost pipe. Ah, is it now? But yeah, kind of wanted to see right here where this, no, just kind of, the strand just kind of ends right there at the edge of this organic matter. Oh, some little round thing in there. But yeah, it's always cool just looking at. Yeah, it almost looks like an amoeba. But yeah, just these pieces of organic matter that are in the soil are always cool looking. Especially when they have strands of fungi and stuff poking in and out of them. That's what we want. We want that good inoculation and endophytes. Endophytic bacteria that goes inside of the plant and is actually digested by the plant. That seems to be an amoeba right there. Looks like a Pac-Man ghost, actually. If you twisted it around at its side. What are you? This could be a fungal spore. That could be a fungal spore. Or just a little piece of clay or silt. But that looks a lot like a fungal spore. That's probably definitely a fungal spore. Yeah, a lot more spores. Good amount of bacteria. We got a Pac-Man ghost. Hopefully he's beneficial. And we don't need yeah, I was hoping to find some Pac-Man spores, because that's supposedly what the arbuscular mycorrhizal fungi spores kind of look like. You know, round like this, but they're usually split open like a Pac-Man. But I haven't spotted anything quite like that yet. But yeah, quite interesting. Kind of want to take a picture of this little ghost here. There we go. Snap the picture of the ghost. Yeah, we're at 400x now, yeah, so we're pretty close. It's a pretty small amoeba, if that's what it is, which I'm pretty sure it is. Usually testate amoebas are shaped like that, but that's pretty small for that type. Anyway. Yep. We got one of these interesting looking. Yeah, thought I figured out what these are the other day, but I think I forgot. So many microorganisms, and the names are hard to remember. I need to start making some notes of what I see commonly, because there's so many different types and forms, and we're still finding more. We have no idea. We don't even know how many types there are. Interesting little strand. Yep. Alright, well, I think that just about will wrap things up for today. So, yeah, this has been sample 3 of our indoor pots here. And enjoy the content, and enjoy learning about microscopy and regenerative soil. Like, subscribe, share, all those good things. And until the next stream, happy growing, and we will see you then. Thanks for joining.